Good evening, Ayman. I'm Rahul Saha, and in the last class we had talked about the ornithophily, the chiropterophily, right? Anemophily, hydrophily, and all that. And we had also discussed or commenced the talk regarding the adaptations or outbreeding devices or the contrivances which promote cross-pollination event. So in that case, we had discussed the first point known as the decliny or unisexuality, where the flowers must be unisexual. That will increase their probability of cross-pollination because they do not have any other sex. So the example were mulberry, papaya, and then we had talked about the dichogamy also, dichogamy. Okay, so in case of dichogamy, I told you that in bisexual flowers, anthers and stigma sometimes they mature at different times. Okay, so there are two types. One is known as the protandry. In protandry, what you have is the anthers mature first. Okay, but the stigma mature later on. So anthers first mature. Okay, then we have the protogeny. Okay, in this case what happens is the stigma of the carpel, it matures earlier than the anther. So here the stigma matures first. Okay, so now we'll be coming on to the suppression of one sex. Okay, suppression of one sex. So in certain bisexual flowers, one sex organ, either stamen or carpel, certain bisexual flowers, you can say one sex organ, either stamen or carpel, is completely suppressed and becomes sterile. And becomes sterile. Okay. Now what happens is anther does not form pollen grains and carpel does not produce ovule. So one of them will be completely suppressed, and this will again facilitate the cross pollination event. The number fourth type will be your prepotency. Prepotency. In this case, what happens in many plants, the pollen grains from a flower, when present on the stigma of another genetically different flower, they can germinate more quickly as compared to the pollen grains of the same flower. Okay, so you can say in many plants, the pollen grains from a flower when present on the stigma of another genetically different flower germinate more quickly, or germinate quicker, you can say, as compared to the pollen grains of the same flower. Okay. Now this again causes the promotion of cross pollination. So the examples pertaining to this prepotency event are grape, pear, and apple. Okay. We have all tasted grapes, all tasted pear, and apple too, which is one of the favorite fruits circulating around, and it is extremely renowned, these three, right? Now the next, coming to the next point, that is known as self sterility or self incompatibility. The fifth point is self sterility or self incompatibility. Now what does this mean? Pollen grains of a flower are incapable, they sometimes become incapable of affecting or causing fertilization even if they are placed on the stigma of the same flower due to mutual inhibition. Okay, they become completely dormant, you can say. They cannot cause effective fertilization. So pollen grains of a flower are incapable of causing effective fertilization even if they are placed on the stigma of the same flower. Of the same flower due to mutual inhibition. So what you have is, you have the example of potato, tobacco, okay, under this specific pathway. Tobacco, okay. Then the next point that comes under the adaptations is heterostily. Heterostily. So the occurrence of two or more type of flowers here in this case. The occurrence, in this case what happens, the occurrence of two or more type of flowers happens which have different length of styles and stamens. Okay. First of all, the occurrence of two or more types of flowers having different length of styles and stamens. Of styles and stamens. Okay. Now what happens is, in the plants like primrose and jasmine, those flowers, what happens is, in jasmine flower, in primrose flower, okay, the primrose is also very famous for omega-6 fatty acid, evening primrose oil, which is extracted from the primrose only. Okay. So in primrose and jasmine there are two types of flowers. Okay. While in oxalis there are three types which are based on in oxalis plant there are three types of flowers. Okay. So these types of flowers are based on the length. solely on the length of their styles and stamens. So an insect visiting a flower, what will that do is, an insect visiting a flower will get dusted with pollen grains on a particular area of its body depending on the height of the anther present. Okay, and then now this insect will do what? Will pollinate only that flower in which the stigma lies at the level of the anther of the first flower. So this process involves pollens and stigma, you can say, from two or more flowers having that similar height. Okay, that is known as heterostele. If they are of different height, that will again promote cross pollination. Okay, then you have the next type known as hercogamy. You had dichogamy, now you have hercogamy. In some flowers, there may be some physical barriers, you can say. This is due to some physical barriers. So the physical barriers may be present in some flowers, right? Between what? Between the anther and the stigma. Why? So that pollination between them becomes difficult. So physical barriers may be present between them, you can say, becomes quite difficult, okay. Or you can say it can or even become impossible. Okay, like you have in case of examples of calotropis. Okay, in case of calotropis, what you have is the pollens are present in a sac-like structure. They are known as pollinium. Okay, pollinium, and thereby pollination is entirely, you can say, dependent on the mercy of insects. Okay, then only pollination can occur. So what happens is, this is a type of, again, a specific way of causing, you can say, or promoting cross pollination event, right? So then we had talked about the double fertilization event already. Now we can begin a new topic and commence a new topic of this chapter known as post fertilization events. Okay, post fertilization event. Good evening, Vanshika. Good evening, Tarun. Good evening, Ashma. Okay, so post fertilization event. This is very, very important. Post fertilization event. After fertilization, what all events take place, or what all events do take place, that is the meaning of post fertilization event. Now we know that we have something known as endosperm formation. How does it happen? Now we know that after the triple fusion or the vegetative fertilization, not the generative one or the syngamy, the vegetative fertilization, where the, you can say, the fusion happens between the male gamete and the polar nuclei. You cannot call it polar nuclei, you can call it a fusion nucleus, right? So having the ploidy of 2n, so it becomes 3n in the end, and now that is known as primary endosperm nucleus, PEN. So the topic is endosperm formation, first thing, because the PEN will then eventually become an endosperm. Okay, endosperm formation. So the formation of endosperm is initiated by, we all know that, mitotic division of the primary endosperm nucleus. So that is primary endosperm nucleus. That is 3n, right? Ploidy is 3n, and this will start undergoing mitotic divisions or mitosis, not meiosis. Keep it in mind. Okay. Now the formation of endosperm occurs when? It occurs usually prior to the zygotic division. Occurs mostly prior to the division of the zygote. Division of the zygote which was produced via syngamy or generative fertilization, 2 and 1. Okay. Now endosperm, it does what? It accumulates food reserves and functions as the nutritive tissue for the developing embryo. Okay. Now what I'll tell you is there are three main types of endosperm which can be classified. Okay, as follows. Three main types. Know it properly. Number one is the nuclear type. Number two is the cellular type. Number three is the halobial type. Okay, there are three types of endospores. You can say or endosperms, okay, which are produced. Now, number one, if you focus upon, is the nuclear type. So, what happens in the nuclear type? Nuclear type endosperm. So, in nuclear type endosperm, what happens is the first division, you can say, a primary endosperm nucleus, 10, okay, which is 3N, and few subsequent nuclear divisions, okay, are not accompanied by cell wall formation. Very, very important. And few successive mitotic nuclear divisions means karyokinesis is not followed by wall formation. Very, very important for NEET examination. Okay, directly will tick here, is not followed by wall formation. So what happens is the nuclei which have been produced, okay, the nuclei which have been synthesized after the karyokinesis, okay, they remain free in the cytoplasm of the embryo sac, and they may also remain free indefinitely. And in some cases in research papers it has also been found that wall formation does take place later on, okay, but the thing is they remain free for some period of time until in some cases wall formation does take place. Okay, so you can write it down that the nuclei synthesized it remains completely free in the cytoplasm of the embryo sac. Okay, and they may also remain free indefinitely or until in some cases it has been seen wall formation takes place. Okay, so this has been seen in research. Sometimes wall formation can take place. So what happens is, if you take the example of coconut, okay, where you have husk as the sclerenchymatous tissue. So coconut, in case of coconut, the cell wall formation of endosperm is never found to be complete. Okay, very, very important. You can say cell wall formation of endosperm is never complete, has never been found to be complete in status. Okay, so this is the example. One more example I'll give you where the exceptions are there that wall formation does take place. You have examples like the phoenix plant. In phoenix plant, what happens? You have the endosperm which becomes very, very hard. Endosperm. Okay. So now we'll come to the second type, that is the cellular type of endosperm. Cellular type endosperm. Now, just judging by the name cellular type, so what you will expect here is cytokinesis to take place along with karyokinesis, right? So in this case, the cytokinesis or the cell membrane or the plasma membrane division, okay, occurs after each nuclear division of the endosperm nucleus. Means cytokinesis follows karyokinesis, you can say in simple terms. Okay, so cytokinesis does take place after each nuclear division, in bracket karyokinesis. Okay, karyokinesis of or pertaining to the endosperm nucleus. It all starts from pen, right? Again, don't forget. So the thing is, the endosperm it attains a cellular form. The endosperm attains a cellular form, not that the nuclei are roaming free, okay, roaming freely. It attains a cellular form. You can say from the very beginning, because first and subsequent nuclear divisions are all accompanied by cytokinesis and then subsequently the wall formation. So first and subsequent divisions are all accompanied by wall formation. Okay, so that is how it occurs. Okay, the example is petunia, datura, might have heard of it. Datura is also used for producing LSD, right? Lysergic acid diethylamide, one of the types of hallucinogens, you can say. So the classical hallucinogen. Then you have the halobial type. Okay, number 3 is the halobial type. Now halobial type means it is nothing but an intermediate between, you can say, the nuclear type and the cellular type. Always remember this thing. Logically, it is an intermediate between you can say, the nuclear and cellular types. And cellular types. Now, the first division always, now understand it very, very carefully. This is a NEET level question. The first division is accompanied by cytokinesis. Always remember, the first division or the first nuclear division, I should be saying, the first nuclear division or karyokinesis is always accompanied by cytokinesis. Okay, that is the plasma membrane division. Okay, but what happens is the subsequent divisions are not followed by, but the subsequent divisions, very important, are not followed by subsequent divisions means subsequent nuclear divisions are not followed by cytokinesis. So now you may be able to comprehend and decipher why is it known as the intermediate between the nuclear and the cellular type, because the first nuclear division or slash karyokinesis is always accompanied by cytokinesis event, that is the plasma membrane division event, but the later on or the divisions that happen after that, known as the subsequent nuclear divisions or karyokinesis, is not accompanied by, always remember, it is never accompanied by cytokinesis, and what happens is they produce free nuclei. Okay, now they produce free nuclei. Now the chamber towards the micropylar end of the embryo sac, if you can recall, the chamber towards the micropylar end okay of the embryo sac is always much larger than, much larger you can say, than the chamber present at the chalazal end. Okay, at the chalazal end. So a large number of nuclei are formed where? Are formed in the micropylar chamber by free nuclear divisions. Therefore, a large number of nuclei are produced or formed or synthesized in the micropylar end, micropylar chamber by free nuclear divisions. Okay, while the nucleus of the chamber, okay, towards the chalazal end, what happens is the nucleus which is present at the chalazal end, since the chalazal end is not that wide, okay, what happens is it is not that large, so at the chalazal end it divides, but it divides to form fewer free nuclei, or in some cases it may not divide at all. Okay, so it divides to form fewer free nuclei, or in some research cases, research papers, it has been found that it does not divide at all. Okay, so the nuclei present at the chalazal end may not divide at all. Okay, that is also a case, if you can take that case into consideration. Okay, so that was the message here. So this is all about your halobial type. The example is asphodelis. Okay, asphodelis species is the example. Now coming on to the embryo development. the next part of our discussion, the next topic will be embryo development. Embryo development. So what happens is after fertilization event, the fertilized egg is known as what? Zygote or oospore. You all know that. So it becomes zygote slash oospore in case of plants. After regenerative fertilization takes place of syngamy. Now, following a predetermined mode of development which is known as embryogeny, it gives rise to an embryo. So embryo has the potentiality or the potential, you can say, to become a complete plant. That is true. Now what happens is usually the zygote divides immediately after the first division of primary endosperm nucleus. Write it down. Usually the zygote divides or commits to division, you can say, immediately after the first division of primary endosperm nucleus. Primary endosperm nucleus. Okay, or sometimes it has also been found that it divides earlier. Very rare case it is. Okay, or sometimes I'm writing it down for your NEET level examination. Sometimes this thing is very important. You can note it down with me that it divides earlier also. It divides earlier, okay, than the first division. Than the first division, you can say, of the primary endosperm nucleus itself, or pertaining to the primary endosperm nucleus itself. Very rare event, because in the primary endosperm nucleus formation theory that I told you that the formation of endosperm is initiated by mitotic division of the pen, and the formation of endosperm, I had told you, occurs usually prior to the zygotic division. But here I am contradicting my statement because biology is all about, you can say, exceptions, rarities, rare combinations. So what happens in some research papers, which is also actually NEET syllabus, it has been found that it divides, the zygote divides earlier than the very first mitotic division. Okay, what kind of division? Mitotic division of the primary endosperm nucleus, which is 3N itself. Okay, so this is a very rare event, mark it. Okay, now what happens is after fertilization, the zygote rests for a period that varies greatly throughout different taxa, from a few hours to several weeks. Again, very important point. After fertilization, the zygote you can say rests for a period that varies greatly in different taxa okay, from a few hours to several weeks. to several weeks. Okay. So you can say that there are no fundamental differences in the early stages of development in case of, you can say, dicotyledonous and monocotyledonous embryos. This is again one more point that is asked in NEET examination. There is no fundamental difference, means observational difference, when and only when, differences I should be saying, in the early stages of development. Which development? In the early stages of embryonic development, right? In the early stages of embryonic development. Okay, when it comes to dicotyledonous and monocotyledonous plants. And monocotyledonous embryos, you can say, to be more specific. Embryos. Okay, so always remember this thing. So the thing is, this specific, these 2 points are also asked in NEET examination. But what happens is, when we delve deeper onto the later stages of the development, embryonic development, then what happens, the differences, they do flare up, they do show up, as the mature embryos of both the dicotyledonous and monocotyledonous plants, they differ really considerably, okay, with discerning differences from each other. That is the point. Okay, so the point is, they do differ when they become mature or at the later stages of embryonic development, but during the first few stages of the embryonic development, the earliest of the stages of the embryonic development, there is no real discerning or demarcating feature pertaining to a dicotyledonous and a monocotyledonous embryo. Okay, you cannot really discern by just looking at them during their early developmental or embryonic developmental stages. Okay, that whether it's a dicot or a monocot. Even a top, you can say, cytologist or cell analyst cannot describe it or cannot really discern the real difference between a monocot and a dicot embryo. Now the first division of the zygote, okay, is usually transverse. It takes place in a transverse plane like this. Okay, this is the transverse plane. So the first division of the zygote usually takes place in the transverse plane. Again, an objective question. Usually takes place in a transverse plane. In which creature? In angiosperm, in angiosperm plants. In angiosperm plants, or I can say in angiosperms. But again, there is an exception. In some cases, the first division may be longitudinal or oblique. In very few cases, the first division could be longitudinal or oblique. That can also occur. Okay, so the thing is, from these, or you can say from this two-celled stage, because the first division has occurred, what you reach is a two-celled stage. Very, very important. Okay, after the comprehension, the first division I'm talking about means first division is mitotic division. Now meiosis won't be happening. Just like you have cleavage, okay, then morula, blastula formation in case of, you can say, eukaryotes, same way you will be having the first division of the zygote. The zygote is the mitotic division. It is never meiosis. It will be an equational division, not the eductional division. So when you have the mitotic division, it is known as, after you can say mitosis, is known as the two-celled stage. Okay, and this two-celled stage, the differentiation of organs, okay, or till the differentiation of organs, the embryo is known as the pro-embryo. Means still organ differentiation happens in plants. Occurs. Okay. So till that point in time, the embryo in plants is known as pro-embryo. Till it occurs, the embryo is referred to as, very important NEET question, pre-embryo. Okay, very sorry, pro-embryo should be. It should be pro-embryo. Okay, not pre, it should be pro-embryo. Very important, you can underline it, that what is pro-embryo. So from the two-celled stage it starts. From the two-celled stage till the differentiation of the organs do occur, the embryo is known as pro-embryo. Okay, very important. You should be knowing it properly. Now what happens is, next is the elucidation, okay, of the development of dicot embryo. Then we'll come to the development of the monocot embryo. Okay, so before going into that, so that is a huge topic, so that will be dealt in the next class. So we'll talk about the polyembryony here. Okay, before going on to the developmental genetics, that is the huge topic, and that we'll cover in the next class. We can talk about a smaller topic known as polyembryony. So polyembryony is nothing but, it may be defined as what? It may be defined as the occurrence of two or more embryos in one ovule. That's all. That is polyembryony. This was noticed for the first time, again a good objective question by Anton von Leeuwenhoek in 1719 in the seeds of orange. Okay, so in a seed with many embryos, usually only one embryo matures, and the rest of the embryos, they do what? They degenerate. They degenerate during the course of development. Okay, now again two scientists come into mind. One is Ernst and one is Snarf. So these two scientists proposed, according to these two scientists, Ernst and Snarf, polyembryony may be true or false. Polyembryony may be true or false, both. Depending on what? Depending upon whether the embryos arise in the same embryo sac or again in different embryo sacs in the same ovule. Okay, again they gave a different classification to polyembryony. So when it comes to true polyembryony, okay, we have various types of true polyembryony. So first type is cleavage polyembryony. So when we talk about true polyembryony, we have something known as cleavage polyembryony. Now what does it mean? It means that it is a consequence of, or it results from the cleavage of the zygote. It arises from the, you can say, it is a consequence of, from the cleavage of zygote. Okay, or you can say earlier stages of development pro-embryo, means two-celled, till the differentiation of the organs, or earlier stages or cleavage of the earlier stages of pro-embryo into, you can say, into two or more units. Two or more units. So the example is, you can say, lobelia, erythronium. These could be the examples. Okay. So the cleavage polyembryony is extremely common in case of gymnosperms, but it is extremely rare in occurrence. When you talk about the angiosperms, note it down very, very carefully. So it is pretty common in case of gymnosperms. Okay. So I'll write it down for you. Common in gymnosperms, and you can say very rare in case of angiosperms. So one more thing I'll tell you for making it very, very clear. Okay. If you still have confusion, false polyembryony is what? So if you have false polyembryony, okay, take out the perflection. It involves fusion of two or more, you can say, nuclei or nucleus pluralis, nuclei, or development of two or more embryo sac. So it involves can say fusion of two or more nuclei, okay, or development of two or more embryo sacs. You can say within the same nucleus. In the same nucleus. And in case of true polyembryony, the additional embryos arise in the additional embryos arise in the embryo sac you can say either by cleavage of the zygote, either by cleavage of the zygote, okay, or from the synergids and antipodal cells. From the synergids and antipodal cells. So this is the true meaning or differentiation between, you can say, false embryony or false polyembryony and true polyembryony. Always remember this specific point of difference. Okay. So remember this point. Now, what we can do is we can talk about point number 2. Point number 2 is under true polyembryony. Point number 2 is you can have embryos from cells of embryo sac other than egg, okay, which I told you, synergids and antipodal cells. So embryos from cells of embryo sac other than egg. So the embryo may appear from synergids and antipodal cells, which I was just telling you in the definition. Synergids and antipodal cells. There's a repetition here, but we have to write it. Okay, so I just correlated that with this definition. Okay, because the concepts remain the same. So the embryo may appear from synergids and antipodal cells in the embryo sac itself. Now the synergids may be fertilized by the male gamete. How? From an additional pollen tube or developed without such fusion. Okay, so that can happen, or additional embryos may appear, okay, from unfertilized synergids also, and may stay haploid in nature. That can also happen. Unfertilized synergids can also give rise to embryo, that is haploid embryo. Okay, haploid in nature. Now embryos from antipodal cells have also been found to form, but they are very, very rare. Embryo formation from the antipodal cells is a very, very rare event. Okay, it's quite rare. So in the next class we'll talk about the other types of true polyembryony, and we'll talk in detail about the development of dicot embryo, okay, in Capsella bursa pastoris, also known as the crucifer type, and also the development of monocot embryo. So these two mechanisms will deal in depth and in detail, okay, in the next class. So I hope you are able to comprehend the concepts taught in today's class, and if you have had any doubts or queries, you can definitely contact me on my personal number to get my prompt reply. Okay then, thanks a lot. Let's meet you in the next class.